started.
Alright, so today we're going to be streaking out plates in a quadrant style. So we have our plate and our bacterial strain ready. And the first thing I'll do is get a loop prepared. So, keeping the tube as sterile as possible, make sure it's clean and safe. Here we're taking a little bit of bacterial ice, and it doesn't have to be a lot because there's a lot of cell on here. Now for the actual quadrant streaking, we will apply the bacterial sample directly on the first quadrant. As you can see here, we'll try to be as liberal as possible for the first quadrant because this will be the most high density part of the plate. Like so, and then I will turn it, also turn the loop a little bit, and then we'll make a second quadrant out of this first quadrant, and this will be our second quadrant. So notice I'm not going back into the first quadrant, so this will be a lighter dilution, kind of fewer cell count, and then a third quadrant which will streak out from the second and into a third, like so. And then for the fourth quadrant, this will be our lowest dilution, kind of a streak method into our fourth quadrant. And this should be our lowest cell count on this last quadrant there. Yeah.
The lighting helps us see the streaks.
Yep. You'll be able to see those four. And that should be, the last quadrant should be where we can see individual colonies. So, alright, thanks for viewing.